Hello, so we did our presentation on noroviruses, which are also nicknamed norovirus, and I'll get there in a minute. So for some background, these are positive ssRNA viruses that are non-enveloped, very small compared to other viruses, and they fall between 7 different genogroups, with G1, G2, and G4 being very pathological to humans, and G1 and G2 being the most common. Uh, Norovirus is the most common cause of gastroenteritis outbreaks in the world, and in 1968, the first known outbreak of norovirus occurred in Norwalk, Ohio, which is why we sometimes call it norovirus. And the R naught varies between 1.2 and 7.2, depending on the population. So it's very contagious and very able to cause epidemics, and it's very virulent. If you look at the diagram on the right, there are roughly 19 to 21 million cases every year. Transmission occurs through fecal oral route. That's the most common way, which causes contaminated food and liquids, and this can occur at all levels of the food system, so from crop to fork. It can also spread through fomites, and another way it spreads is through aerosols, through vomit, and it's also nicknamed on top of norovirus the winter vomiting virus because it's more common in the winter since people tend to be at home longer and transmission is more common. So for pathogenesis, it affects the tough cells of the intestinal tract and also the stomach, and it causes these cells to proliferate and induce an immune response for eukaryotic parasites. So it's obviously the wrong immune response, and this is why we see some of the symptoms of the virus. And these cells also tend to be immunoprivileged, so the virus will proliferate in these cells and stay in the cells for around 8 weeks, which is why we sometimes see it for 2 months instead of like 2 days, and this is why it's very commonly spread. And for symptoms, patients will usually see the symptoms between half a day and 2 days. And like I said earlier, it causes acute gastroenteritis, which is nausea, severe diarrhea, abdominal pain, and it's not out here, but vomiting. And children under the age of 5 and the elderly are the most at risk of this virus.
This outbreak occurred in November of last year in Colorado. Hundreds of students and staff in Mesa County Valley School District 51 had been complaining of an illness that comes on incredibly fast and leads to vomiting and diarrhea. 30% of students and 20% of staff called in sick at a high school. This is where and when the outbreak began. It seemed to be contained to just the one high school, which had about 1,000 students, until a week later it started spreading to other schools in the district. The school district had about 22,000 students and had never had to close all their schools due to illness before, and this was because of a norovirus outbreak. Noroviruses are one of the most infectious viruses known. Usually, up to 70% of the people have projectile vomiting, which is full of the virus and helps spread it around. When it comes to schools, it's always a good idea to disinfect desktops, pencil sharpeners, and water fountains. Those seem to be hotspots for norovirus and also germs in general. Also, a little bit of extra context of norovirus outbreaks in general. Um, from August of last year to March of this year, there were 920 norovirus outbreaks reported by Norostat participating states, which is about 12 states kind of spread throughout the US. So that's just kind of to let you guys know that outbreaks of norovirus happen all the time, especially in schools in the healthcare setting. The outbreaks tend to happen in the winter months as well, and they happen throughout all of the US states.
To the general public, it may seem that antibiotics may seem like a treatment. However, they only treat against bacteria and other strains, so this will not kill the virus. Currently, there are no vaccine treatments for norovirus. However, rehydration with water is the only therapy in suggestion by the CDC. If necessary, worst case scenarios for those who cannot retain body fluids, they must be admitted into a hospital. Typically, patients with norovirus will begin to see a sense of relief within 1 to 3 days after staying home from recovery. Lastly, it is important to detect as well as inform the public about how and where this virus can go. One detection that has been primarily used in labs for both molecular and medical research is the complement of RT-PCR and droplet digital PCR, With RT-PCR, we get set results to not only unwind its DNA and add set primers, but since norovirus is an RNA virus, we have to use reverse transcriptase to go from RNA to DNA. From this, using primers and probes which are specific to norovirus, we can actually generate more virus during the extending stage of PCR, also known as polymerase chain reaction. This can be very vital and important when diagnosing a patient. On the other hand, droplet digital PCR partitions our samples into 20,000 droplets and has been shown to be more accurate than RT-PCR. Stool samples are another way we can diagnose a patient as well as detect, as stool samples can present any inflammation in our patient. To the general public, the CDC provides links to organizations such as National Outbreak Reporting Systems, CaliciNet, and Norostat. These all help regulate and detect viruses monthly.